Assalamualaikum. My name is Nurul Ifa binti Ahmad Kamizi. My student ID is 2024276042.
Hi, Assalamualaikum. My name is Akila Anani binti Saiful Anwar and my student ID is 2024260672.
Hi, my name is Raja Sofia Batrisha binti Raja Saiful Izan and my student ID is 2024691064.
Hi, my name is Nurulaika Yasmin binti Zarini and my student ID is 2024444318.
Hi, my name is Siti Roshavika binti Muhammad Hanif and my student ID is 2024441946.
Before we go further, let's get to know about analytical chemistry. So, when we talk about analytical chemistry, it is all about analysis. So, basically, this involves developing and applying method and high technology instruments to obtain precise information about the composition, structure, and nature of substance. So, in other words, analytical chemistry is like a detective in chemistry. There is two types of analysis in analytical chemistry that falls into two major branches. The first branch is qualitative analysis. We use this type of analysis when we want to identify the chemical elements or functional groups are present in the substance. The second one is quantitative analysis. This type of analysis being used when we need to determine the amount of percentage of those identified components. Just now, we understand the different branches of analytical chemistry. Now, let's look at how this science functions in the real world through forensic science. Forensics is the perfect example of applying this natural and digital science to legal investigations. Historically, courts relied on subjective witness stories. But now, we have modern forensics that use empirical method to detect, analyze, and interpret the scientific data to find objective proof. And the question is, how do we actually get that proof from a piece of physical evidence? The answer is, we can use the techniques in analytical chemistry called classical technique and instrumental technique.
So, what is chromatography? Chromatography is an analytical method for separating and identifying a mixture constituent part. It functions by enabling the component of a mixture to pass through a stationary phase with the aid of a mobile phase. These phases will move at varying rates and separate because various substances interact with them in different ways of a mobile phase. In order to identify and purify chemical substance, usually chromatography is frequently used in scientific research, drug detection, and medicine. The second one is principle of chromatography. So, the technique is based on how substances have varying affinity for a stationary phase and a mobile phase, which then it will causes each component to split and move at various speed. The first phase is mobile phase. Mobile phase is the phase that moves through the stationary phase. It can be a liquid or a gas, and then it carries the mixture along the stationary phase, and usually the components that are more strongly attracted to the mobile phase will move more quickly. And then the second one is stationary phase. Stationary phase is the phase that remains fixed and does not move during chromatography. It may be a solid material or a liquid supported on a solid surface. The components that are more strongly attracted to the stationary phase will move more slowly. Due to these differences in movement, the component of the mixture will become separated. Thus, the separation will allow us to see the component to be identified, analyzed, or purified.
Okay, so next we go with the importance of drug detection in forensic science. Do you guys imagine if in the crime scenes, how we know the white powder is whether sugar, flour, or an illegal drug? So that's why drug detection comes in. So drug detection is important because it helps forensic scientists identify unknown substances accurately. It also helps investigators solve drug-related crimes such as drug trafficking and possessions. Besides that, drug testing can be used on biological samples like blood, urine, and hair to determine whether someone has taken illegal drugs. Most importantly, the results can be used as scientific evidence in a court, which is helping judges make fair decisions. So, drug detection plays a major role in both crime investigation and public safety.
Legal settings.
Okay, let's move on. Do you guys ever wonder what the theory of forensic drug detections? Firstly, the basic idea is that different compounds move at a different speed. When a sample enters the chromatography system, the compounds begin to separate based on their chemical properties. As shown in the flow diagrams, the process starts with the sample collection and preparation. The sample is then injected into the chromatography system where the compounds are separated in a column. Next, the separated compounds pass through a detector which produces a chromatogram. So basically, chromatograph is like a fingerprint for chemicals because each drug creates its own unique patterns. By comparing this pattern with known drug standards, forensic scientists can identify the drug present in the sample. This makes chromatography a reliable and powerful tool in forensic drug detections.
Urine test is one of the most commonly used methods for drug detection because it is simple, cost-effective, and non-invasive. When a person consumes a drug, the body metabolizes it into chemical byproducts known as metabolites, which are excreted through the kidney and can be detected in urine samples. The procedure typically involves collecting a urine sample in a sterilized container and analyzing it using screening tests such as immunoassay. Positive results are usually confirmed using more advanced techniques, including gas chromatography, which mass spectrometry, or using liquid chromatography, which tandem mass spectrometry. Urine testing also can detect a wide range of drugs and is widely used in clinical, forensic, and rehabilitation. Next, we have blood testing, which is it detects drugs and their metabolites directly in the bloodstream, making it one of the most accurate methods for identifying recent drug use. It is because drugs enter the bloodstream shortly after the consumption, which blood analysis can provide information about the concentration of a substance in the body and help to determine whether an individual is currently under the influence of drug. This method is commonly used in medical, forensic, and legal settings, although it is more invasive and expensive than the urine testing. Saliva testing, which is also known as oral fluid testing, involves collecting a sample from inside the mouth using a swab. Drug present in the bloodstream can pass into the saliva, allowing recent drug use to be detected. This method is increasingly used in workplace screening and roadside testing because it is quick, convenient, and difficult to tamper with. However, saliva testing generally has a shorter detection window than urine or hair testing and is the most effective for identifying recent drug exposure. Analyze a small sample of hair, which is usually taken from the scalp, to detect drugs and their metabolites that has been incorporated into the hair shaft through the bloodstream. As the hair grows, traces of drugs remain embedded within the hair structure, providing a long-term record of drug use. Hair analysis is particularly useful for detecting repeated or chronic drug use over several months. It is commonly used in workplace, forensic, and legal settings. However, it is generally less effective for identifying very recent drug consumption because drugs require time to become incorporated into growing hair. Sweat testing uses a sweat patch attached to the skin to collect sweat continuously over a period of days or weeks. Drugs and their metabolites excrete through the sweat accumulate within the patch and can be later be analyzed in the laboratory. This method is often used in rehabilitation programs, probation, monitoring, and research study where continuous monitoring of drug use is required. Although sweat testing is less commonly used than urine or blood testing, it provides valuable information about drug exposure over an extended period. There are few cases in drug detection that have been searched, which is detection of lisdexamfetamine in a forensic investigation in 2023. In a forensic autopsy case, investigators test the urine and stomach contents of a deceased person to check for drug use. Using the liquid chromatography, they detect lisdexamfetamine and its metabolites D-amphetamine. The result helped forensic experts determine whether the drug contributed to a person's death. This case showed the importance of drug detection in forensic investigation.
Let's discuss the advantages of chromatography in forensic drug detection. First, chromatography offers high sensitivity and trace detection. This means it can identify extremely small amount of drugs, even when only trace residues are present on clothing, surfaces, biological samples, or evidence collected from crime scenes. Second, chromatography is considered the gold standard for reliability in forensic laboratories. The technique provides accurate and reproducible results, making it highly trusted by forensic scientists and acceptable as scientific evidence in court proceedings. Another important advantage is its ability to distinguish genuine drugs from counterfeit drugs. By analyzing the chemical composition of a sample, chromatography can determine whether a pharmaceutical product contains the correct active ingredients or has been illegally manufactured. Chromatography also has broad applicability across different types of compounds. It can be used to detect a wide range of substances, including narcotics, stimulants, prescription medication, and their metabolites. In addition, chromatography provides rapid analysis and high resolution. Complex mixtures can be separated efficiently, allowing scientists to identify multiple compounds within a single sample. Some chromatographic systems are designed to be portable for a field use. These instruments allow preliminary drug screening at locations such as crime scenes, border checkpoints, and airports, reducing the time needed for laboratory analysis. Finally, recent developments have introduced AI to enhance chromatographic analysis. AI algorithms can assist in peak identification, data interpretation, and pattern recognition, improving accuracy while reducing human error. Chromatography also has several limitations. One challenge is low analyte concentration. When the amount of a drug present in a sample is extremely small, detection becomes more difficult and may require additional sample preparation or concentration steps. Another limitation is signal suppression. This occurs when other substances within the sample interfere with the target compound, reducing the detector's response and affecting analytical accuracy. Chromatographic analysis can also be affected by background noise. Unwanted signals from solvents, reagents, or instrument components may interfere with the detection of compounds and make the data interpretation more challenging. Lastly, overlapping peaks can occur when two or more compounds have similar retention times and are not fully separated. This may complicate identification and quantification, especially when analyzing complex forensic samples. In conclusion, chromatography is one of the most powerful analytical techniques used in forensic drug detection. It transforms invisible chemical information into reliable scientific evidence. Whether identifying unknown substances, monitoring drug abuse, detecting counterfeit pharmaceuticals, or supporting criminal investigations, chromatography provides accurate and dependable results. As a result, chromatography remains an indispensable tool in forensic science, helping investigators uncover the truth and ensure